Chapter 3 is all about enzymes, a biological catalyst. We will start off with 3.1, the mode of action of enzymes. First of all, I'm going to start off by giving you a formal definition for enzymes. So if a question asks you define, describe or outline what an enzyme is, this will be your formal definition. Enzymes are biological catalysts. That's the first part. You have to specifically mention that they are not just any form of catalysts, they are biological catalysts. I'm going to be underlining the keywords. They are biological catalysts that increase the rate of reaction. Okay, that's the next key point. They increase the rate of reaction by reducing activation energy, and providing alternative reaction pathways. Let me clarify. Alternative reaction pathways. And one of the most important things when it comes to an enzyme is that they cannot change after the chemical reaction is complete. They should remain as it is, so they remain unchanged in a reaction. Okay, let's dig into each part separately. Enzymes are biological catalysts, so they are of a biological origin. What is the first thing that an enzyme does? It increases the rate of any reaction that's going on. How is it doing so? It's doing this reduction, it's doing this increment of rate of reaction by two pathways. Number one, it reduces activation energy, which is the minimum energy you need for a reaction to go through. And the second way is it provides alternative reaction pathways, meaning if you wanted to take A to C, let's say this takes 10 seconds. But if there was this alternative pathway where A could be converted into B and then into C and it only takes 3 seconds, what are you going to prefer? Are you going to prefer this pathway or are you going to prefer this pathway to obtain C? Obviously the fastest pathway, right? So that is exactly what enzymes do. They know instead of doing this, we can do this, so let's do this. That is an alternative reaction pathway. Okay? The most important thing, no matter what, this enzyme, typically this is how I draw enzymes, like Pac-Man. Okay? After the reaction is complete, Pac-Man has to remain as it is. It can't change. It cannot be converted to something else. It can't be used up by something else. That's not possible. And if you recall about proteins, you will remember that according to protein structure, hydrophilic groups will always be pointing outwards, right? That is the exact same thing for enzymes. If you remember, enzymes are tertiary proteins. So we can say that all enzymes are globular proteins with their hydrophilic R group. So hydrophilic, what does hydrophilic mean? Hydrophilic means water loving, with the hydrophilic R groups on the outside. Thus, enzymes are indeed soluble. Enzymes have to be soluble for them to catalyze metabolic reactions. So a variant of enzymes would be something that goes along this spec. The enzyme can be either intracellular or the enzyme can be extracellular. What does intracellular mean? Functioning inside the cell, obviously. What does extracellular mean? Whenever we have an enzyme that is extracellular, it is functioning outside the cell. No big deal. There are two main mechanisms in which enzymes actually work. Now, this is the interesting part. First of all, this was the older model, and we called this the lock and key hypothesis. Lock and key hypothesis. These were models that were used to explain the functioning mechanism of enzymes. First of all, we had the lock and key hypothesis, the lock and key mechanism, but with time we improved, we updated ourselves, and now we have something known as the induced fit method. So the induced fit hypothesis. If these things make no sense to you, that's perfectly fine. What are we trying to do? Let's understand what we're trying to do. What we are trying to do simply is we're trying to understand how an enzyme works. We're trying to understand how an enzyme works. So for that, I think I'll go with a yellow. Okay. This is what happens in the lock and key hypothesis. I'm going to give you some statements first, then I'm going to give you a visual cue to hold on to. Whenever it comes to the lock and key hypothesis, what's going to happen is that the substrate is going to fit into the active site. What in the world is the active site? We'll talk about that in a bit. Here, the substrate is going to fit into the active site of the enzyme. That's the first thing. After the substrate has fitted into the active site of the enzyme, let's see what happens. After that fit occurs, we say that the only reason why it was able to join is because the substrate is complementary to the active site. Substrate is complementary to the active site of the enzyme. So I'm going to move induced fit down a little bit and let's see. So this is the idea. If I have an enzyme like this, and let's say I have a this shaped something, here's the deal. So that is our overall enzyme. This is what we refer to as the active site. This is where the substrate, this thing, goes and binds into, like this. Okay? Whenever we talk about the lock and key hypothesis, we have to say that the substrate, this little thing, is complementary to the active site. It's perfectly complementary to the active site, meaning this is a box, that's a box, box can fit like that. Box can fit like that. That's the idea. The substrate is complementary to the active site of the enzyme. Just like so. That is the lock and key hypothesis. So then why exactly are we talking about it in the form of a lock and key? That's because we're simply referring to the enzyme as being the lock and the substrate as being the key. We need a perfect fit for it to work. If you try opening your door with a different key, it's not going to work because there's no perfect fit. That is why we call this the lock and key hypothesis. But why is it outdated? Why do we use a new hypothesis now? Let's see what the induced fit model talks about. Now, the induced fit model says that we have a substrate which is almost or partially, almost or partially complementary, almost or partially complementary, that binds to the active site. What does that look like? Let's see. Now I have the enzyme. I'll use the same shape. And now I have the substrate, but this substrate is not a perfect fit. There's a small hitch to it, like. So if this was the lock and key model, this guy cannot fit into the active side because it's not perfect. You see, there's this little hitch. It has to be a perfect fit. It's not a perfect fit. There's this unwanted hitch here, right? If this was the lock and key hypothesis, this would not happen. This substrate and this enzyme will not happen. But the induced fit hypothesis, the induced fit model simply says that even if there's a small difference in the substrate, meaning even if the substrate is almost or partially complementary, it can bind. It can bind. Okay, so let's bind this and see. Now, okay. Now what's going to happen is the enzyme will adjust itself a little bit to perfectly fit into this substrate. So if I get my eraser and if I reduce the size of it, this active site will change itself a little bit and inducedly fit the substrate. Now it's a perfect fit. Now it's a perfect fit. So if I were to word that, this is how I would say it. The substrate which is almost or partially complementary now has bound to the active site. Therefore, the active site changes shape. It undergoes some kind of change to, here's the deal, better fit the substrate. It's trying to better fit the substrate and catalyze the reaction. And catalyze the reaction. Catalysis occurs, reaction goes on, rate of reaction is increased. Those are the two models in which enzymes are described as being put into work.